So in this lab, you're going to need grapes, or you can use blueberries or something similar. But you need to find 5 that are the same size. If you have a kitchen scale, you're welcome to use that to weigh them, but that's not necessary. Don't go out and buy one if you don't have one. You can definitely just size them up. Just make sure they're the same. So we got those 2. And you're also looking to make sure there's no damage to your grapes. No like premature cracks or indentations or anything like that. So once you have your 5 grapes selected, set them all to the side, and you can either label your cups if they're like plastic cups you're not going to reuse again, or you can write down what each cup is. So for example, in the first cup, I'm going to have just water. So I would label water, and then you're going to have your cup. You're going to fill it up with the same amount of solution, so you're going to control that variable. Maybe it's going to be like half of a cup or something. So fill up your cup, and you're then going to add your grape. Now, you're going to let this lab run for a few days. So today it's kind of premature measurements and making hypotheses. You're going to predict what you think is going to happen to this grape. In this cup, do you think it's going to be an isotonic solution to the grape? So do you think the grape will stay the same? Or do you think it's going to be a hypertonic solution to the grape? Do you think it's going to cause it to shrink and lose water? Or do you think this solution's hypotonic to the grape? Do you think this is going to cause the grape to swell and gain water? So you're going to go ahead and take any measurements you want to. Some people use a piece of string and or a roller, and they measure the grape that way. Other people just take a picture now, and then they compare the picture in 2 or 3 days. So you go ahead and place this grape in the cup, and you'll do this for all 5 solutions. The next cup I'm going to use is sugar, and I'm putting 2 teaspoons of sugar in my cup. So we're going to add half a cup of water, and we'll mix this up, and then we'll add your grape. Placing this grape in here. This is my sugar cup. And then we have the same amount of salt in this cup. So we have 2 teaspoons of salt. So we're going to write down cup 3 is going to have salt, 2 teaspoons. And we'll just label this cup 3. And again, you're going to add half a cup of water. And you're going to stir that up. And we're going to add our grape. And it's okay if your solids aren't fully dissolved into your cup. The next cup we're going to make is a fruit juice solution. So we've got our half cup of water, and we're using this Mio strawberry kiwi. We'll just do 2 good squirts. It's probably about 2 teaspoons. And we'll just label that as Mio. And then you'll be adding your grape in. In my last cup, I'm going to actually be using coffee. All right, we've got our last cup here, and we're going to be adding our grape. So we're going to let that sit for a couple days here, and we've got our labels as to what these are. I'm actually going to have to put mine someplace else to keep it safe from my children. But keep your label nearby, keep them in the order that you set them up as, or at least write a number on the cups. And if you can't do that, then you can just use a piece of paper and just lay it under the cups and write it right under the cup. But this is our first setup of our labs for today. So I have these here. The next lab you're going to possibly do, if you have the availability, is using gummy bears of any sort or some kind of gelatin-based snack to do a controlled experiment. So this one we don't have a control, but if you did want to have a control, if you had an extra grape, you would just leave that grape in an empty cup. I don't have another cup right now, but you could use this grape to compare to the other grapes in a few days. It's not exposed to the solution, so it's not exposed to the independent variable. I'll show you the setup for the gummy bears now. Alright, for this experiment, we want to use all of the same color fruit snack. And you need 4 of them. So I'm going to use the red ones. I don't have enough of the other colors. We are going to have 1 cup of water. In this experiment, I highly recommend using cold water. So we're going to do 1 cup of water here. And the gummies all are made from molds, so they're going to all be the same size, but you're welcome to measure that with a ruler or take a picture of them. And then you're going to place your gummy in the cup. And then you're going to set up your sugar and salt. They should each have about a teaspoon of sugar and a teaspoon of salt in your cups. And we're going to add our cup of water to each. And you're going to need to stir these. Once you get that situated, you're then going to add your gummy. And then your 4th gummy is going to be used as a control. So you'll be able to compare this to your gummies in the salt and your gummies in the sugar. And you can run this experiment for 24 hours or 2 days or 3 days to see how your results are. Make sure you label these cups, or at least write a code for them so that you remember this is salt and this one is sugar. So I'm just writing SU for sugar. And then you'll be able to write out your lab report. So you're going to type out your hypotheses, write out your materials you're using, write out your procedure that you're conducting this with, record your data, make a graph if you feel like it, discuss your findings and your results. Then you're going to conclude. You're going to restate your hypothesis, accepting or rejecting that, and explaining why. I also want you to try to use the words hypotonic, hypertonic, and isotonic in this experiment.